Hi, I'm David Liew from Melbourne, Australia, reporting from Room Now from ACR 21, the virtual conference. And I'm delighted to be joined by Seoyoung Kim, who is from the Brigham and Women's Pharmacoepidemiology Group, and is a rheumatologist there as well. Seoyoung, welcome to Room Now.
Sure, thank you. Thank you for the invitation, and delighted to be here as well. And I'm in Boston, so pleased to meet you, and whoever is listening later. Thank you for listening.
And congratulations as well on your ACR award. You won the Henry Kunkel Early Career Investigator Award, and we have a lot of people at Room Now who were rooting for you. So congratulations on that as well.
Thank you. That it means a lot, and it's such an honor. I've been, kind of hoping and wanting to get the award ever since I was a fellow. So kind of like my dream came true. It's a really good feeling, and I am very grateful for that.
Well, I mean, I think we're all very glad your dreams came true, and also that I think it's reflected in the whole body of work that you presented a whole lot at this meeting, but in particular, we're here today to talk a bit about the STAR-RA study, which looks at the topic of the day, Tofacitinib safety.
Yeah, so of course, Tofa's trial data are somewhat, I think, unexpected, and it was, I don't know, like disappointment to some degree, and I think a lot of patients were also surprised. I was surprised, and because I think it's a priori, I don't think anybody really kind of thought that would happen, and I've been following a lot of safety clinical trials just because I like studying drug safety, and we don't really have the luxury of having safety trials for every single drug. So whenever something like this happens, I get particularly excited because this is when I get to have the gold standard data from trial for the drug safety, and then I get to use real world data to see whether that is the case in more generalizable population, right? So for me, it's a very unique and important opportunity, although what we found or what they found is somewhat unfortunate. But in any case, I think as you know, the oral surveillance trial was done because of the regulators' mandate. Phase 2 and 3 TOFA trials, they had some concerns about certain safety signals, and usually phase 2 and 3 trials are too small, too short to find anything meaningful with regard to the safety risk. So obviously this phase 3b4 safety trial was initiated many years ago, and this is an issue with all the trial, right? So one is very expensive. Two, it takes time to get going, and even after they start enrolling, it takes time to finish all the enrollment and follow up. So I think it was started many years ago, more than 5, I think probably 7, 8 years ago, and now we are still waiting to see the final product of the trial, because we heard a lot from Pfizer during this meeting, basically, the piece of different outcomes, but we still don't have the full paper. While, initially, I think maybe more than a year ago, the first signal they found was a VTE risk associated with a high dose of tofacitinib, 10 milligram BID. And then, my group at the time, there was last ACR, actually, we presented at the late breaking, I think, or maybe 2 years ago, I don't remember. But we actually conducted the risk of VTE associated with TOFA compared to TNF, not knowing that's what they were going to find, because mainly we conducted that study based on the the signal from Baricitinib trial. So in a Baricitinib pre-marketing trial. And then while we were conducting it and we were presenting at AACR, the FDA communication came actually afterward. So we somehow happened to be at the good timing, and our work was published in Arthritis and Rheumatology at the time. And then, for cardiovascular risk and cancer risk this time, we got the FDA announcement and some other media news release earlier this year, I think February 2021. And then we immediately wanted to run a real-world based cohort study to replicate and to kind of see, their validity in a different data source and in a more representative population. But what we actually did, and we didn't probably get to talk about it during the meeting, was we actually registered the full study protocol at clinicaltrial.gov. So if you search clinicaltrial.gov with a STAR-RA study, you probably find our protocol with all the details, just like, the trialist register their protocol at clinicaltrial.gov. We actually, even though we are not required to do so, we wanted to also be as rigorous as possible. So we registered our study protocols upfront and then initiated our analysis. Using 3 data sets, we wanted to, ask the same question, whether the risk of cardiovascular event is higher or not higher with the TOFA compared to TNF in the overall RA patient. So that is our primary cohort. We call it real-world evidence cohort. So everybody, I think age 18 or 20, I think that was our adult age cutoff. And we had all 5 TNF as a class. So if they are taking one of them, they're in the reference group. And then for the subgroup, out of this real-world evidence cohort, we had the RCT duplicate cohort. So we can mimic as much, again, not exactly because we don't have ACR criteria, we don't have certain things that they have in the trial, but we use some of the criteria that the OR surveillance trial had for enrollment to restrict the study cohort for a high cardiovascular baseline risk. And then ran the study, we made sure the confounding was minimized. We cannot be completely avoided, but we've done our best to minimize. And what we found is, again shared during this meeting, in the real-world evidence cohort, we found a comparable risk of cardiovascular events between TOFA and TNF inhibitors. But in the RCT duplicate cohort, again, patient with a RA and some baseline risk factor for cardiovascular disease, we found a numerically increased risk of cardiovascular disease similar to what OR surveillance trial found. Our patients are older, generally speaking, because we had a Medicare data set included. But I think what this tells me is, if I have a patient who is like 40-something, 50-something year old, female or male, but have no other worrisome risk factor for cardiovascular disease, maybe their absolute risk of cardiovascular disease from TOFA is maybe not that high. Maybe that's okay. But if somebody is like over 60, 65, or even 70, and they're a smoker or had a small MI before, something like that, then maybe those are people, either I try to use something else, or if TOFA or is the only thing that I have, then I have to have a serious conversation, right?
Absolutely.
So I think that's what we found.
Yeah. Well, congratulations on such a rigorously run study. Do you think that the results that you have from STAR-RA have made you believe the concerns that have come out of oral surveillance more or less? Do you think that that's really kind of strengthened do you think that helps to validate it, or do you think that it actually adds a different perspective?
I think it kind of confirms, I guess most of the time, usually clinical trial confirms observation studies. I'm a little careful to say that our study confirmed the trial, but I think our results, and it's the duplicate part, is in line with what the trial showed. If you get really picky with the statistical significance, we can go on and on, but I'm not a big p-value fan, so point estimate is in line with their point estimate, and it is, I think, really interesting. And we don't have a full, and I think the thing that people are bothered by is we don't have a good explanation why this is the case, right? We don't really know why tofa is more harmful in certain patients. There's really no good mechanism. So that's a little bit annoying, because we usually want to know why this is the case, but we don't have that answer right now. But I think our result is, for bigger population, somewhat reassuring. For high risk cardiovascular population among RA patients, I would be very cautious.
Well, thanks for giving us that perspective on essentially what seems like a two-track approach that we have to take to tofa nowadays. Thanks very much for joining us today, Seoyoung.
Thank you.